Hello, everyone. Thanks for being here. It's a pleasure to be here. I'm Daniel Moreira. I'm Brazilian. I am an assistant professor of computer science at Loyola here in Chicago, so I'm local. Welcome to Chicago. I hope you're enjoying. Hopefully, the weather will be better tomorrow. Sorry about this. Yeah. And yes, this is a work that I did together with my collaborators from Brazil, Dr. Rocha from the University of Campinas, and now Dr. Cardenuto, who was just defended. And yeah, I'd like to share with everyone our findings characterizing problematic images. So just to make clear to everyone, as I mentioned, we are all computer scientists and we approach the problem from CS standpoint. And at the top row, this is how our labs approach scientific integrity. We focus on developing software tools and doing CS research and development where we want tools to support authors, reviewers, editors, and investigators to foster and verify scientific integrity. And the way we do it, if you look at the bottom, we rely on machine learning, we rely on data-driven solutions and AI, what means that we need training data. We are crazy about Retraction Watch and PubMed, to be honest. And yeah, what we do, we take all this data that we collect, we do manual annotation, we invest a lot of time on these. And this work is our effort to understand the landscape, and because we have limited time and resources, we want to figure out what are the priorities we should follow. And I want to share with you the numbers, the quantitative analysis we've made. So just to make it even clearer, we are experts in image processing, image digital forensics. And I did my PhD on this topic. So we have a focus on analyzing the images, if they were tempered or they are pristine or generated by AI. And yes, I'm pretty sure you know that several of these misconduct practices then involve uh, doing wrong things or manipulating images. We have several of these cases. Reuse that is not acknowledged, tempering, and even AI generation. These are the sources we use it, so I just wanted to share with everyone. We started with Retraction Watch, as I just mentioned to you, that this is a good starting point for us. It's easy for us to process their data. We took a snapshot from up to the end of the past year, 2024, and we collected 56,000 entries of actual retractions. And from these retractions, we filtered only the ones that involved the images. This is our main focus. So in the end, we focused on 8,000 of these entries. And then because many of these retraction notices, they are not very computer friendly, they don't give us enough information for organizing data in the way that we can feed our machine learning solutions. We relied on pub-peer and manual reading on going through the cases. So we matched Retraction Watch and pub-peer discussions, and we filtered from the 8,000 entries to 2,200 of these entries. And because there are several, not several, but some papers there are actually involved in more than one entry, we removed the duplicates, and in the end, we had around 2,000 articles that we processed objectively and quantitatively. So we are using this fancy word characterizing, so I just want to be clear what is it for us in terms of CS standpoint. And to characterize the misconduct, we used three perspectives or 3 point of views. The first one of the three is by looking at the image types. This is important because there's no way, or it's very challenging to develop a single machine learning model that will be able to deal with all the different types of image. So it's advisable that we focus on particular types, and we want to understand the landscape, so we made a study to understand. Just to be clear with everyone, when we say image types, this is what we are talking about. So it's understanding that there are figures that are published on the papers. There are gel blots, there's microscopy, colony, tumor, transwell, wound sealants, and others. Actually, for this, we used the taxonomy that was inspired by a just recent paper that was published by Dr. Brooks. And yeah, when we counted the number of image types from the actual retractions, these are the numbers that we got. And I'm sorry, that is a little bit maybe small, but we had a predominance of gel blots. That's the first one that you find here. And that's no surprise, actually. This is a trend that most of us know. 51% of the cases involved this type of images. The second one was transwell and so on. So we have these distributions. It will be useful for us maybe to figure out what should be our priorities and what our solutions should target. The second point that we analyzed in terms of perspective was in terms of manipulation type. And it helps us again because machine learning won't be able to focus on all the types of misconduct operations. So we need to organize them somehow and then have different solutions or different methods for different manipulation practices. And for these, we also use a taxonomy that was based on Dr. Brooks' work. And we had an organization also from our media forensics point of view. And when we're talking about manipulation types, these are the manipulation types. So for example, one of them is replication. That's when you simply reuse an image without acknowledging it properly, no changes. Splicing is when you compose an image from two or more other different images. Recoloring, that's when you reuse an image and just change the color scheme. We have flipping, rotation, several operations, and even we have one case where there was AI generation. That is something that we work with and have tools for detecting. And in the end, yeah, by looking at the cases until 2024, this is the distribution. 87% of the cases involved replication or reuse. And the second one was resizing. And yeah, it might be related to how easy these practices are to do. But yeah, I'll leave this for the discussion that we're going to do later. And then going to the third perspective that we took that will help us, that has been helping us to give priority to our approaches. We organized things in terms of problem context. It helps us to understand for the tools that we are developing how they should operate, in what level, what order, how they should, the mechanics, how they should we process PDFs, extract the images, and then look at them, things like this. And for this, we organize it in 3 groups. So on the left, we have single figures. These are the cases where by analyzing a single figure, taking it as input, it's enough for you to figure out if there was a problem. We have situations where the manipulation is within the figure, the single figure. So if you think about figures that are published in papers, they are often multi-panel. So you have like figure 1A, 1B, 1C. So these cases are panel only, a single panel, and copy move is one example where folks just replicate pixels to hide things or maybe to highlight some things that they like. The other case is within figures, you have different panels but the same information. These are where you process single figures. For multiple figures, we have situations where you find reuse of the figure with some changes. This may happen within the same document, within the same manuscript, the same scientific paper, and also it might exist across different papers. So one image has been reused across different papers, so we organized also in these terms. And finally, we focus on paper mills. Yeah, from the past 5 years, it became a major thing. And everything happens in paper mills at scale. This is automatic. It doesn't follow the past practice where everything belonged to a same culprit that was doing on several of their papers aspects. Now we have these crime ring organizations. They're just generating several of these papers, and they sell authorship, so authors are not related. We have a paper on this and plus one for detecting some interesting results. So I advise to take a look. So these are the numbers we found. Most of the cases, 59% is between papers, so it's reused between papers of images between papers, and that gives a nice perspective for us. Yeah, so as just to wrap up here, as I have the last minute, discussions and next steps. We found by looking at these cases that there is a prevalence of problems with gel blots and between paper image replication. So images are simply replicated across different papers. And one thing that we find challenging is that retraction notices, they do not provide enough information for us to have a proper annotation, understanding, and then generate ground truth. We have to go after other sources. So maybe we need to think in a more comprehensive way to provide the retraction notices. And I'm being selfish here, I'm a computer scientist. That could help us to do the computation on feed our machine learning solutions and train them. And just to publicize to everyone, we are now in an effort where we just collected actual images from retracted papers where we did copy move manual annotation. So these were practice of copy move or cloning of regions. We annotated the regions manually, we developed a tool for that. And we are now in contact, just signing documents for contract and stuff. We will be working with IEEE and Kaggle, and there will be a challenge that folks will be able to subscribe and submit their solutions, and we're gonna rank their performance, and hopefully we have some money to give to the first positions. So this is what I had for today. I'll be happy to take your questions, and thanks for your attention.
Hi, I'm Nihar Shah from the Machine Learning and Computer Science departments at Carnegie Mellon University. Really wonderful talk and wonderful work. My question is, like, given the advancements in diffusion models, like this is going to be an increasingly challenging task, right? So what do you think about an alternative approach where like we use public key cryptography to ensure provenance of each of these images? So if you might know, like FDA CFR 21 Part 11 already has e-signatures in it. And I think at least Thermo Fisher has already implemented it. So it's available there. So should we as a community just go all in on just ensuring like provenance?
Yeah, that's a great question, and I know there are some efforts, including I think Adobe, where they're trying to include these IDs or these authentication codes in the images. Yeah, it's a matter that we need now to, if you're gonna follow that path, I think it's worth it. And I don't know what happens when you compress those images or when you recapture and things like this, but this also meets the quality, because maybe you should really like sharing the raw image. That's the bigger advice. But yeah, we still need work. I know research is going on, and I can see solutions are possible, and we need to do even better work, like sharing this with everyone, and making it intuitive for everyone to use outside of CS, and then making this maybe a requirement. Okay, we will only have an image on your paper, and we're gonna only have the raw images. We went through these authentication processes, but it's definitely a valid way of doing research on authenticating these images. Yes.
Uh, Thanks, Jim Taylor, Cleveland Clinic Dermatology. Appreciate your great presentation. I was wondering about image flipping. Have you noticed any issues related to risk factors for wrong site surgery? I would premise that question by pointing out that we've seen this with patient-submitted images. They get flipped. There was also a software program that we were using that actually accidentally flipped an image. And then when you're looking at get images in Epic, you can actually flip the image. But when you close the program, it goes back. And then some surgeons use actually image flipping as part of their procedure. So that complicates the issue. But it's a potential problem, I think, for wrong site procedures, especially like dermatology or plastic surgery.
Yeah, so finding flipping is actually an operation that is, from the CS standpoint, of course, easy to detect. And to be honest, I've seen situations of papers that we had a publication in Springer Nature a couple of years ago. We processed some retractions, again, some images, and we found, this is a very famous case of this guy that just he reused an image across different 8 papers. And we found the information that was in the retractions, but the flipping ones were missed, even for humans. So even for people, it's hard to find. And it was flipping gel blots, by the way. But the solution that we have for provenance that we use, it's a image processing one using key points. It is able to detect flipping, so it's possible. And to be honest, flipping is one of the things that we do for training the machine learning solutions. So it's in the repertoire, but yeah, we need to work better to make this available to everyone, and everyone understand that it's possible to detect. Thank you.
Hi, yeah, Valda Vincent from the Science Journals. Really nice work. And interesting because you're looking at retractions where presumably the problems are serious. So I'm interested though, whether you were looking at all at context. So which figures were duplicated? Was it sort of a core central figure of the paper? Was it a control? Was it, and I'm asking just because we actually check figures and too often see duplications of things like actin controls, where there's absolutely no reason you should have duplicated it, right? And so I think there's there's a higher amount of sloppiness than there really should be, and there's too much sort of putting the wrong figure in the wrong place because your figures weren't well organized. So I just want to know the extent to which you think mismanagement is a problem, you know, as opposed to misconduct.
Yeah, you're right. Especially when you think that some of these papers, PDFs, they might come from LaTeX, some code, and then you may just point to the wrong figure when you compile the PDF, and then you're gonna have the duplication. Yeah, the tools that we develop, they detect them irrespective of intention, and this is something that we always defend. We are not like pointing fingers at everyone. We're just bringing evidence, and then it's the part of the editorial process or investigations to rule what's going on, maybe a request for correction and everything. You have a great point about the control, because from the CS standpoint, we are not experts in these. And to be honest, we don't take these things in general into consideration. And this is where we should increase collaboration. And to be honest, I'm looking forward to collaborate with everyone, because this is the type of knowledge that I need, because I could include these in our pipeline for the solution and then have a better result, or a more useful result.
I mean, the reason I think it's so important is because we focus a lot on trying to prevent misconduct, which is absolutely correct. But I see much less focus on trying to teach people to manage their data, which needs to be done.
Yes, and to be honest, as I presented, it's not only for supporting investigators or sleuths or yeah, also the authors, right? So if you have this tool that is supporting you, and we will point out, hey, by the way, you have the same image and you shouldn't, like an assistant that uses these tools, then I can avoid this problem while I'm writing, and then I can also supervise my collaborators' work. So this is where I'm basing my research at this moment. I'm looking forward to develop tools in this regard. This is how I'm, by the way, basing my tenure track. Hopefully, I'll get there. Yeah. Thank you.
I'll just ask you one question.
Sure, please.
For a within image, an image is provided. What do you think your sensitivity is to determine whether that image has been manipulated in any way?
So it depends on the nature of the image. For natural scene images, media forensics is full of solutions that you check the noise of the pixels, like local noise. And when there is a composition, the noise of regions, they will not match. And then you can find regions that are not, they do not follow the same distribution as the others. But for scientific images, take, for example, GeoBlots, grayscale, tiny, resolution, not that great. These solutions do not automatically transfer, and they perform poorly. And folks have confidence that I don't recommend in media forensics that everything will work for scientific images. I don't think it's the case. We need to improve and adapt to this image. So bad. I would say the results will be bad. Yeah, for the tools that we have, we need to do better.